a shared node, the RAS-ERK cascade, is involved in many different, say, growth factor and stress responses. Yet you can have different growth factors that lead to different fates, even though they use that same molecule. And so how is, if that node gets activated, how does the cell actually know what decision to make and what outputs to have, or what it was that caused that? And so there are many different, there's several different models or extreme models of how this could work. Of course, there's evidence, quite a good evidence that in some cases this occurs through combinatorial encoding, that you could have actually one particular receptor might activate this common node, but also activate pathway-specific branches, and that these together are interpreted to give you a different output. And there are examples of things like where RAS and the PI3 kinase branches of pathways downstream from receptors act together to give specific pathway-specific outputs. Another idea that's been out there for quite some time is the idea that there could also be dynamic encoding or dynamic filtering. That is the idea that even though you activate the same node, the temporal pattern by which it's activated could specify unique information if different inputs generate different sort of patterns. And of course, there's a lot of beautiful work, including work from Sylvia Santos and Philippe Bastians, who's going to talk next about the dynamic patterns that you see of RAS activation, RAS-ERK activation with different growth factors, and how these seem to be correlated with distinct responses. So what we thought is that one thing that would be interesting to do is to ask, we now, instead of looking at these systems in these other ways, could we actually get optogenetic control over the RAS-ERK module, and then now probe directly how the cell interprets different patterns of RAS activation. So how did we do this? Again, through a very simple sort of idea. Jared looked for molecules that could activate RAS, but were completely dependent on recruitment. And so we found that the catalytic subunit from the SOS GEF is sufficient if we recruit it via the 5-PIF interaction. So it's really acting, 5-PIF is acting much like, say, GRAB2 would act downstream from the receptor. So if we do this, we can actually get RAS activation. We can follow this very easily in live cells with a labeled ERK molecule. This ERK, once phosphorylated, will go into the nucleus. We can see when we hit it with 650 light, the activating light, it goes into the nucleus, and we can track it through different cycles of turning on and off RAS. It responds very quickly. Key thing here is that these are live cells. We have a live perturbation, live cell perturbation, as well as a live cell readout. So we can do this single cell living cells. Jared also looked at what were the outcomes in terms of just following things like phosphorylation of AKT, which is downstream from PI3 kinase, as well as ERK. Both of these are activated by Western blot if you stimulate with, say, PDGF. But if you hit them with this light, this octo-SOS construct, what you see is you get only ERK activation. You don't see PI3 kinase AKT activation. Even though those are thought to be downstream from RAS, it looks like it's not sufficient. So it looks like we are really specifically activating the sub-network that exists below RAS. Okay, so I'm going to tell you about two general things that we tried to look at. One is to try to look at the flow of information between RAS and ERK. Then we have this perturbation and this readout. Can we actually quantitatively characterize how efficient, what's the nature of how information is transferred from RAS to ERK? And then secondly, I'll talk about our efforts to try to ask how the cell interprets different dynamic patterns of RAS-ERK activation. Okay, so as I said, we have a cell where we are able to look at this module, the RAS-ERK cascade, and we can use light to control the node directly above it, and then use optical methods to measure the activity of the node right below it. And we actually can directly measure not just how much light, but of course there's a lot of cell-to-cell variability in terms of expression. We can actually look directly at how much of that SOS cat is recruited to the membrane. So we have a very precise measurement of what the input is. And so we can, in principle, get this kind of transfer function or dose-response curve, if you're a biologist, that looks at how the fraction of nuclear ERK changes as a function of how much of that SOS cat you have at the membrane. Okay, so this is one of those experiments. We're hitting it with different cycles of light. This is sped up about 10 times. But we're hitting it with light. You can follow that, see the sauce cat gets recruited, and then you get ERK localization to the membrane following that. You can change the dosage of light, and then thereby change the dosage of recruitment of sauce cat, and then get curves. And we get curves for each of these individual cells. So these are what some of the dose response curves look like. Now, one of the major questions in cell signaling now is really how precise can a cell be in terms of detecting information? Is it really able to, in a very analog way, be able to detect subtle differences in input and then make result in subtle outputs, or are they sort of more binary in their nature? And if you look at the population of cells, and most of the methods we have to look at this kind of thing are population-based methods, you see that there is a lot of noise here, and it doesn't look very reproducible in terms of the dose-response curve. But as you see here, these are the single-cell dose-response curves that we're able to generate now, and they're far more precise. These are some of the distributions you see for 2 example cells. They are different from one another, but they all, both the amplitude and exactly where the threshold is, but they all have approximately the same Hill coefficient, and they're remarkably precise. We can actually go up and down these many times over the course of many hours of experiment, and we still always get back to the same point that, on this curve. So it's very reproducible, very precise, and also this happens to be reversible. There doesn't seem to be any memory or hysteresis, at least when we look between the RAS and ERK set points. So the other thing that we wanted to look at is to look at different type of experiment, which is frequency response. This is a common engineering sort of thing to see how efficient is this particular pathway at transmitting signals that have different dynamic characteristics. So we can put in, ideally, different frequencies of input, and then measure output and ask, how is that signal transformed? How is it degraded? Do you see a lower amplitude? Do you see a phase shift? And we can ask, how does the gain, the efficiency of transferring the amplitude, how does that change as a function of measuring it at different frequencies? And there's been a lot of beautiful work on this sort of thing. For example, Alexander van Oudenaarde has done this on the yeast osmolarity pathway. And when you look at sort of a physiological response like that, what might we anticipate? In that case, we see something that's this bandpass filter, where that means that there's an optimal frequency at which for transfer of information through there. And so in the case of a biological function like the osmolarity pathway, that frequency, the optimal frequency, is determined by this adaptive feedback structure of the network that controls what the optimal frequency of stimulation is. So we might anticipate that if we look at this pathway, it might show that kind of bandpass behavior, a preferred physiologically relevant frequency. But it could also be a low-pass filter, something that just is able to process slow, long signals, but maybe filters fast ones. It doesn't pass very fast, kind of noisy signals. Or it could be an all-pass filter that's very sort of agnostic about what the frequencies of the timescales of the information is. So Jared did this, and he actually didn't do this by scanning through different frequencies, but he actually decided to stimulate the cell with a mixed frequency light, and then he could measure also the sauce cap recruitment, as well as ERK output, but then Fourier transformed this data in order to generate an entire frequency spectrum here. So we're looking at input frequency and then what the sauce to ERK gain is. So this is what the curve looks like, and what really, we were a little surprised by this, because again, we were kind of anticipating more of a bandpass like, where there were important selective frequencies. But what we found is that actually, interestingly, there certainly is a range here below of a high frequency. And so I'm going to refer to frequency, but I'm also going to talk about the lifetime of the signal. You can think of that as what's the sort of the pulse width of the signal that would be effectively transferred. So what is happening here is that signals that are shorter than, say, 4 minutes are effectively filtered. They don't really pass efficiently through this pathway. But once you get up past 5 minutes and go all the way out to many hours, the efficiency of transmission is more or less equal for all of these things. So it looks like it approximates, in some sense, except for this range of very fast filtering, more of an all-pass filter.